So next speaker is the keynote speaker, Maria Chikina from University of Pittsburgh. She's going to talk about biophysical sequence model, sequence to function models.
All right, well, thank you so much for inviting me. It's really a pleasure to be here. I've really enjoyed the conference so far. And okay, I'm gonna start with like a little interactive thing here. So we're on the West Coast, and but last year on the East Coast we had a solar eclipse. Who went to see it? Wow, lots of people, right? It was awesome. And we were able to do this because we got this really nice map that tells us like exactly where you have to be in the zone of totality. I had to drive quite a bit to get to it, but it was totally worth it. And really, like, we didn't think too much about how that happened, right? So what is the first time somebody in the human history was able to correctly predict an eclipse? Who thinks it's like before Common Era? Okay, who thinks it's like modern times, last 500 years? Okay, so actually you're both right. It just depends. I didn't phrase the question correctly. Like it was not a well phrased question. So it turns out like a lunar eclipse people actually were able to predict like before year one. And the solar eclipse really was modern times. And so the thing is, there is this 18-year cycle that the three celestial bodies, like the Earth and the Moon and the Sun, have periodicity in, and this was discovered a long, long time ago. And both like the solar and the lunar eclipse obey this cycle, but for whatever reason, one of them we were able to predict, and the other one not. And so what is the reason? Well, this is the reason. It's just when the lunar eclipse happens, everyone can see it. Everyone for whom it is night. Right? This is where you can see lunar eclipse. This is the path of the solar eclipse. And so just the precision required to predict this is much, much higher, even though they kind of obey the same cyclical behavior. And so perspective, like of an ancient observer that doesn't go very far from where they were born. The lunar eclipse happens roughly every 18 years, so you would actually observe it multiple times in your lifespan, and like you could develop some kind of pattern matching in your head about when the next one is going to happen. From the perspective of the solar eclipse, it happens roughly every 400 years. So by that time, like your culture has turned over and you've lost all your records, right? So, and that's kind of where the problem, the prediction problem gets really hard. So this is actually the first instance of somebody correctly predicting the solar eclipse. And it was Halley of Halley's Comet, which he's somehow more famous for. I feel like this is actually really, really impressive because this was he published this map of the zone of totality before it happened. Then there was another map that was published after it happened, and I didn't put it up here because they're actually like, they look the same. It's absolutely incredible. But he had to use like calculus and the laws of gravitational motion that were pretty recent discovery. And so to kind of like put all of this in perspective, right, we have our ancient astronomers. They're doing pattern matching, and they're getting some kind of correct predictions. But really, it's pattern matching. They have no idea about physics and even like basic geometry. And then we can have kind of these what I call laws for special cases. So like Kepler laws for predicting planetary motions, assuming everything is ellipse, and these work really well. But the orbit of the moon is actually not an ellipse perfectly, and that's enough to throw the whole thing off. And in order to make these kind of predictions, we really needed we needed universal laws and this concept that you have to like integrate them over time to make predictions into the future. So why is this actually relevant? So I mean, we're also in the business of making predictions, right? We have lots and lots of prediction problems in computational biology. The one that we heard a lot about today is predicting things from sequence. And really kind of what we want to predict is kind of how each cell interprets its DNA. So there's some kind of common germ in DNA. Each cell sees something very different in terms of its chromatin, and this dictates all of the functional output and ultimately the phenotype of the cell. So the prediction problem we're trying to solve is like predict the output of the genome conditioned on the cellular context. So we have this like specific function f like for this cell and a different function d for this cell. I just want to linger here for a little bit because I think this captures what we're doing right now, even though in theory there's actually some kind of universal function. And I think people are moving in that direction, and I think it's going to be really interesting to see what people do there. But for now, a lot of this is kind of like context conditioned. And so, so we're interested in these sequence to function models, right, where it takes sequence and we predict it using usually some kind of neural network. And so this is informer predictions, right? So we have this is the measurement, this is informer predictions. These look really amazingly the same. So we're like, wow, this model has learned biology, right? And I guess to a certain extent it has, but then not to belabor this point again, but in a different perspective, it actually didn't. And in some sense, this is actually we're just not, on the precision scale, we're just not where we want to be on the precision scale. We have some amount of precision, we just don't have all of the precision. It's just these personal genome effects are tiny relative to the other effects that you saw in the other plot. So you can just think about it like, well, we have kind of like a Kepler model, not a Newton model, right? So I guess kind of why is gene expression really, really hard? So maybe lots of answers to this question. This is kind of the one that I've settled on. Is in some sense we actually have like a fundamental data problem. There's only so many genes in the genome, and the input for the gene expression of a gene is actually really long, right? So you have these regulatory elements and maybe very, very far away. So you need to consume like all of these different, so much sequence, most of it is irrelevant, find all the relevant parts and synthesize it into the output. And maybe it's possible that actually every gene locus is unique. And like, okay, what do I mean by that? That's kind of a strange thing to say. But maybe there's millions of biomolecular interactions possible, right? And maybe there's, for like a particular gene locus, there's one that really matters in terms of precision that is completely ignorable for every other locus, right? This is like consistent with what we know about biology, and there are like some very special loci that people listed data in detail, and it's like, I don't know if that happens anywhere else, actually. And so this is just to argue that like maybe it makes sense. Like this is the like once in 400-year solar eclipse. Like maybe like the simplest thing to do is actually develop some general, like not wait for more data or like more compute, but actually develop some kind of like generally applicable methodology that's going to improve precision. So I'm going to tell you about like really tiny baby steps we're trying to make in this direction in my group. This is kind of generally in the sequence to function modeling space, and we've heard really amazing talks today about like just completely incredible like engineering and data efforts and people like so many GPU hours, right? Like so much work and really amazing results, like incredibly promising stuff. And so I'm going to tell you about basically none of that. We're doing like really simple things that like run on my desktop and are very easy to compute. So it's a little bit just the opposite. So I'll tell you about this work that we call the total interval sequence to function model, which was in ISMB proceedings from last year. And I'll also give you an update on model we presented. I had a postdoc presented last year as a short talk, the differential loop extrusion model. Don't quite have a preprint, but it's coming. It is, like maybe like a month. OK. All right. So for the totally interpretable sequence function model, we're very much inspired by this work from Sarah's group. So often we care a lot about like the predictions that our model is giving and how accurate we are. Is like your architecture better than my architecture? Oh, I got like an extra like 0.01 whatever like performance. But we also actually, a lot of times we use these models for just biological insight, right? And I think this is a really nice paper that really highlights the capacity of these models to deliver that. And so in this paper, they were looking at predicting chromatin accessibility across cell types and really focusing on the contrastive aspect, like really looking at like what makes the cell types distinct. And they had kind of a scientific objective, not like a building blocks predictive model, but the scientific objective was like, let's try to understand what makes these cells distinct. And the architecture is not the most complex architecture, but it was one that was optimized for the scientific objective that they were interested in. And so this is AITAC is actually it's a model and a kind of interpretation framework. And so what it does is it takes convolutional kernels and it maps them to motifs. So you can take these interpreted as motifs. Everybody has done that. That's no surprise. And you can track these motifs to the final layer activations through filter ablation. So after you do this, you get this kind of, it just gets basically a transcription factor to cell type map. And this map contains a ton of biological information, and we can kind of spot check it for correctness. So for example, we have like PAX5, EBF1 for B cells. This is immune cells. We know a lot about how they differentiate. So you can do a lot of spot checking. So we have like ROR-C, ROR-gamma for LC. So all of this makes sense. And from this, we can maybe trust that some of the other predictions they're making are also actually correct, which turned out to be the case. OK, so this kind of gives us like a roadmap for how to interpret these models. And so what exactly we do, right? So we have the first layer convolutions are interpretable as motifs. That's pretty straightforward. But it's not a single layer network. We have these other layers. What do they do? We know we need them for prediction. It makes a big difference when you put them in there. What do they do? So well, you can kind of hypothesize of all the different things that they could potentially be doing. They probably do all of these things. And so the really kind of fun and like answer from the perspective of biology that I think we all get excited is that they do some kind of like combinatorial modeling, right, where transcription factors have to work synergistically to regulate chromatin structure. But I guess there are also what I would call like boring answers potentially. Right, so you need extra layers to do some extra motif calibration. So these kind of like right, so we have like these kind of PWM motifs. We know they're actually not accurate. The independence assumptions, like people have demonstrated, those are not actually 100% accurate. So you could do some more calibration on that. And you can also do some calibration for just like binding probability, because the motif just gives you a score. It doesn't give me binding probability, which is actually the unit that I would want. And also, OK, let's say they do actually convey some kind of TF-TF interactions, which I think definitely does happen. It's really hard to kind of tell from the network exactly what kind of interaction is it computing, right? We can think about kind of a perfectly linear interaction where the combination, like we just like linearly add the motif activations. And in fact, we saw with Puffin, like that's actually like very effective for some tasks. Like linear seems to be good. And then we can also have these nonlinear kind of functions, but we can have both like synergy and saturation. I think we get like very excited about synergy. Saturation seems really boring. I mean, I think maybe we would like to know which one of those it is. So I guess the question that we pose is like, instead of trying to figure out how to like implement a really sophisticated post-hoc analysis that's going to answer all of these questions, why don't we just put a model already in this form, where it was just going to like intrinsically answer all of these questions with its parameters. So this is the model. We call it totally interpretable sequence to function model. And basically, it's still kind of like a neural network architecture in a way, but it doesn't have any uninterpretable hidden layers. So every layer is either DNA or it's transcription factor activation. And the way we went about constructing the layers is, it's inspired by what we know about biology. So I'll just quickly take you through how we did that. All right, so we have this top layer, which is just a kind of like a very basic convolution with some kernels, and we actually initialize them to known motifs. We can fine-tune it later. And so we just perform a convolution. We get this like very basic motif activation score at every position. There's a kind of a nonlinear calibration for each activation. It's trying to map it onto this probability space, which is where we want to be. And then we actually pool everything across all positions with trainable pooling. And the trainable pooling, basically what it does is it can model both like an average and a max. And we had actually pretty specific hypotheses for what we thought the model would do. So transcription factors vary like really widely in terms of their specificity. Like you can just look at the PWM, like the information content of that PWM, like how many bases is it actually making contact with. And so like a low specificity transcription factor kind of like, it doesn't, there's not like a particular site where it's present with like probability 1, right? It has kind of like these on-off dynamics, and it can bind lots of different places. And you certainly don't want to do a max in this kind of situation, because it would just lead to a ton of false positives. Like your binding site could be so low specificity that literally like any sequence has a match, right? So a max really doesn't make sense. And then we have transcription factors that actually have like extremely specific sites. So like for CTCF, you see a CTCF site, it's like very likely there's a CTCF there. And for those, maybe actually max is sufficient, but we don't want to make these decisions like arbitrarily. We're going to let the model decide, and we can check that later. And finally, we have an interaction layer. And basically, this interaction layer is just every transcription factor gets like kind of a tuning parameter. It's like between 0 and 1 that where the tuning parameter is a function of the presence of the other transcription factors. And that actually explicitly parameterizes this like and or additive contribution where like 0.5 is actually perfectly neutral. If like all of the interaction coefficient is 0, you get 0.5. And so that's the model. And then through all of these steps, you actually maintain TF identity. So every layer in this network, all the rows they have names, right? They're just whatever transcription factors we used to start with. And so the final output is actually just a linear prediction for open chromatin activity. And it actually is immediately interpretable as a TF to cell type map. And just before I show you the results, I just like to point out a few things about the data set that was used. So we're actually using exactly the same data set as the AI attack. And it's a really amazing data set where we have data across an entire developmental tree of immune cell types. And, but it is classified in 7 main cell type lineages. And generally, as like we see and was reported in the original paper, the lineage prediction is much, much easier than the within lineage. Like you can just kind of visually assess like a little, I zoomed in on a little heat map from the original paper. Like if you just focus on a particular lineage, there's all these places where it seems kind of the predictions I'm making are constant even though the data has variability. So, so for most of the talk, I'll just focus on the lineage prediction tasks. And so the way we kind of, we actually assess these results is we assess them as improvement. So we compare like a convolutional tower, exactly the same one that was used in the paper, though we retrain it with our folds, like so all of the training is exactly the same. And the way I report the results is just improvement over a convolutional tower. And so we can see that, in fact, the full model does improve over the convolutional tower. And we have kind of this ablation study where we take out different kinds of different layers from the model. And it seems that the interaction is actually making, our interaction layer is making the most difference. And we can now, the final layer coefficients is just like, that's it. That's what they are. I can just immediately plot them. Nothing else has to be done. They just already look like this. And the results are extremely similar to what you get with AI tag post hoc analysis. For example, B cell, we got literally the exact same result. And similarly with ROR gamma. OK. And all right, so let's look at some of the other parameters, right? This is the final layer. What about some of those internal layers? So for the trainable pooling, we had this extremely specific hypothesis that it should kind of track with motif specificity. And in fact, it does. So this is the information content, and this is the pooling parameter that is like learned from scratch and goes between average and max. And they're very well correlated, though not perfectly so. And we can also look at the interaction layer. This is just kind of a global view of the interaction. So we can look at kind of the final layer coefficient. So this is what gives you all the cell type specificity, just aggregated over cell types. And we can look at the total contribution to the interaction layer. And so one thing to notice is that these are actually quite different. So we have like different TFs that are high for interactions and then don't contribute to TF specificity. And so a really key one here is CTCF. That makes sense because it's like a broadly expressed factor. It doesn't, it doesn't determine cell identity. All cells have it. So, and this is a kind of a fairly general trend where a lot of these factors were not necessarily expressed in every cell type, but just generally broadly expressed. These were the most interacting ones. One thing I said is the way we parameterize the interaction layers, we try to kind of explicitly capture, like, do we have like and or linear? And so just kind of like it's kind of color matched. We have blue would be saturation. There's a negative coefficient. And this was maybe at first a little bit surprising. It's mostly blue. right? Especially like something like CTCF has like ton of interactions, and they're just all blue. So basically, it's capturing saturation. Maybe a little disappointing, but actually, in retrospect, it makes a lot of sense, and it has a very simple kind of biological interpretation. So if I'm trying to predict open chromatin, I see like a strong CTCF site. I don't care what else is there. Like that is already highly informative. Right, and I just want to kind of go back to the full prediction problem with the full tree. So I'm not going to talk too much about that. There's more results in the paper. We did actually, at the end, we tried to use all the data. And I think we did do kind of like an interesting thing here that in general is just, I think, like a good technique for trying to deal with these multitask problems where the amount of, like, there's certain contrasts in the multitask problem, they're like way easier than others, right? And how do you make sure that you're actually finding like the subtle interesting things and not just like getting better and better at doing just the lineage prediction? And so in this data set, there was actually like a pretty natural way to transform the problem that made it more interesting. And so we made a new prediction task, which was child versus parents, so, right? Like you're trying to predict the transition across the lineage tree. And then you're still using all the data, right? So you don't have to like somehow selectively throw out the data or make kind of like a new data set, but it's just been transformed this way. And I just like to point out, like, even though we have these kind of parent-child relationships here, this is always possible because you can always like make up a parent by just taking an average. And this way you can form a bunch of kind of interesting contrastive tasks. And so it turns out, yeah, OK, this is really hard. So basically, everything that's colored is a place where we had a good prediction, and that is just very few edges on this tree. So really hard to make these kind of parent-child predictions all for a few places. And as you might expect, they correspond to like major lineage transitions. And interestingly, doing it this way does, like, all of your B-cell predictions, turns out they are all concentrated in a very specific developmental stage. There was one exception here where we actually were seeming to get quite good predictions for this transition. So double positives to CD4 and CD8. And I don't know, if you've ever looked at like data from naive CD4 and CD8 cells, they're incredibly similar. I mean, very, very difficult to distinguish, like with like single cell, like ATAC-seq or RNA-seq assays. Like, not so much once they're activated, but in naive state, they differ very little, mostly by the expression of the CD4 and the CD8 genes. This is really subtle. But it turns out not only were we able to predict this well, we actually found like the correct biology. Won't go into too much detail here, but for this transition, which has actually been kind of studied in depth. So I want to kind of summarize this with like all the things that we tried, we didn't work, because the model is really simple, and I know like all people are like, why didn't you do this? Why didn't you do that? And the answer is probably like, we did do that, and it just didn't work. And so one particular kind of really major criticism, which still bothers me, and this is something we're actively working on, is this is not a grammar model. This is the billboard model, right? So we at some point, like at a very early point, lose all positional information about where the motifs are, and that is not because we didn't try to make it a grammar model. So what I mean is it doesn't work, because certainly like we have these like convolutional towers, and those are grammar models by their construction, and they do work. So what you're saying, it didn't work. So we kind of had like some different metrics for what we consider working. So obviously performance, right? That not too difficult to make a grammar model work just in terms of performance. We had another metric, which is parameter stability across folds. Now, that is more difficult once you start making the model complex. And finally, we had our most important task, which is how well did we recover, just without any post-hoc analysis, just in the final layer from that final heat map, how well do we recover in the known cell type specific transcription factors? We actually had quite an extensive list because the system is so well studied. And so when you make the models more complex, like these just go away. Your performance will increase. But the interpretability and the stability, which actually go hand in hand, because the interpretable model should ideally return very similar results across training folds, right? If it's finding out something real. So those tend to go away pretty rapidly. So still working on kind of trying to resolve that and make more complex interpretable models. I think this is actually totally possible, and we're making progress towards that. So, all right. So this is that was a totally local model that just looks at like a very small window and tries to predict open chromatin. And of course, it's just like a small piece of the regulatory code. And ultimately, what we're interested in is gene regulation, because that is what is going to give us phenotypes and diseases, right? And gene expression is really complex. It's much, much harder to predict. We've heard many talks about that today. And it's so complex because just so many things have to come together. And you have all these distal elements that come in contact with the promoter. And this is like a very complex process that's orchestrated over like mega-base scale. So we have some data that kind of tells us a little bit about what's going on at the correct scale. And so we have these kind of high C, micro C data sets that capture some of this 3D folding that we think is important for orchestrating gene expression. And so this is an example of, hopefully this is familiar to everyone, this is an example of a high C matrix. So where does the high C data come from? So I think when people first looked at this, there were maybe quite a number of theories about what is actually going on at the molecular level that is giving rise to these patterns. And so when you obviously we observe these kind of diagonal square structures, which are now are known as topologically associated domains, or TADs. And one thing you can imagine is like a TAD is like just kind of like a bundle of DNA that is like all in close proximity in the cell, but that is like a model that mostly we've moved away from. And so now what we understand is happening is that this pattern is orchestrated with what we call a loop extrusion process. So instead of this being the picture in one cell, in a single cell, actually what we have is a bunch of different states that just in aggregate produce this. So the loop extrusion hypothesis hypothesizes that, I was going to say it's a hypothesis because actually not everyone believes it. But I mean, so the idea is that you have the cohesin complex, which is going to bind at some particular point, and it's going to start like here. And it's going to start extruding a loop, and the loop just progressively gets longer and longer. And the cohesin has two legs, like a left and a right, and then they can both move. And the rates at which they move is variable across the genome. So when you see like a boundary like this, that is kind of a control point where the cohesin leg seems to get stuck and not move across that position as efficiently. So the important thing is to realize that what we now understand is the contact map is like, yes, it's an average across cells, but a really important aspect is actually an average across time. That even if you had like a super homogeneous cell population, you're still somehow not seeing the full picture. So we've been working with kind of this idea. So our model is actually very heavily inspired by this kind of like 1D loop extrusion simulator by Jeff here. And so this is a really simple model that models this process, but it never instantiates like the 3D chromatin structure. It actually just adds context to the contact map. So the way we can look at this model, so we have like a cohesin, and at first it binds, and it binds right like at the same location. So it binds somewhere on the diagonal. So it's making a contact very close to the diagonal. So just maybe like a kilobase away, and at any point it can just maybe do nothing. One of its legs can move forward, or it can completely detach. So now if you simulate this process, and for all of these like time steps, you just add a add a count to the contact map, you get something like this, right? So the cohesin moves along. Sometimes it just stays in a particular position a little bit longer, so you get more counts, and it like kind of randomly moves its right and left legs. Sometimes it gets stuck because these are actually convergent CDC outsides, so it's really stuck on one side here for a lot. It only moves in one direction, and eventually it falls off, right? So this is one cohesin complex. That's its lifespan on this piece of DNA. So now you can add another one. So now there's 2, and you see like they actually get stuck at 2 different positions. And you can do this a lot, and then like really a lot, and it starts to look like a high C map. Okay. So what we decided to do is actually just consider just the steady state, right? So this is, like factoring out all these stochastic processes which are known what happening. Like, what is it going to look like when you just take the average over all time and all of the cellular states? And this is what we call the differential loop extrusion model. And basically, we formulated as the concentration. We consider the concentration in a particular cell, which corresponds to a particular loop. It's just related to how much things are flowing into the cell, which is like how much of the loop is being extruded, and how much things are flowing out of the cell, plus the term for the cohesion coming off entirely. And so, right, so this is now just like we can actually get like this completely closed form expression in terms of these quantities, and this now is not a stochastic thing. It's just a closed form expression for the concentration of cohesins in the cell. So that's why it's differentiable. You can fit it, and you can fit it to data. And so before I show you how we fit it to data, there's kind of a subtle, like seemingly completely kind of pointless discussion, but we actually had very long and lengthy discussions about this aspect. So probably you're familiar kind of with looking with high C on high C in 2 different ways. So this is like the raw data. It's actually log transformed, so you can visualize it a little better, some of these like interesting looping features. And this is the diagonal normalized view. So so we just subtract the overall diagonal value, so it's 0. And this is why you actually see blue is negative, right? You're just like, what does it mean to have negative contacts? Well, it's relative, like it's a log-fold change. And so, yeah, right, how do we fit the data? What is more interesting? So in some sense, the raw data is clearly like, we're trying to fit a biophysical model. Raw data is the physical model. Contacts cannot be negative. Right, but on the other hand, most of the variance here comes from the diagonal decay. And so your model is going to work really hard to fit the diagonal decay before it starts doing anything else. Like it's not going to do any, like many iterations, it just won't do anything interesting. On the other hand, in this model, we see like a lot of really cool structure and like promoter-enhancer interactions. We see them right away. So we do actually work in this space. And the way that we do this, so our model is a biophysical model. It outputs like contacts, it outputs concentrations. So it kind of runs in real units. And just before we compute the loss, it self-normalizes to this view. And I'm not going to go into a ton of details for why this is true, but it turns out that this has like a really nice side effect where we don't have to worry about some of these like terms that come from polymer dynamics, because like we actually have no polymer dynamics in our model. We just have the loop extrusion, and modeling those is actually quite hard. Okay, so this is the DLEM model, and so for we have some kind of initial concentrations in the diagonal, and actually like in reality these are all constant, but you can think about them like potentially variable. And starting with these initial diagonals, we have some parameters that describe how likely you are to move left or right at a particular location, which is these p's, and we have like a off rate. And so we can just go diagonal by diagonal and calculate all of the other values. This is a completely closed form. You can do it for the entire, like if I gave you these parameters, you can just fill out the entire contact map. So one thing you notice is this gets like really ugly really quickly because it's actually fundamentally recursive. So you get these very ugly equations. Another thing I wanted to point out, just like when people are thinking about architectures or what architectures like are biologically meaningful, which is a totally separate question, like what architectures work well and are trainable, right? But if you think about what architectures are biologically meaningful, this is not a transform, this is a scan, right? This is like every computation is actually totally local computation. So in fact, right, there's this closed form for the entire thing. You can like fit it directly to the data. It will work, but it is slow. It is not super stable. So there's actually a better way to fit this. And so this is, I'll just tell you a little bit about how we actually fit this. So we think this is actually now the data, and so we're gonna, the decay parameter, we don't worry about it because everything's diagonal normalized anyway, so we're just gonna fit the left and right slowdowns, which tell you how likely it'll go left and right. And so we can just look at the perspective from the perspective of like a single square. So we're going to make a prediction for this particular square. And this is going to be made in terms of the inflow to the square, which depends on these kind of slowdown parameters for moving left and right, times kind of the concentration of cohesins at the previous looping step, at like the slightly shorter loop. So that's the numerator. And the denominator is just the outflow, is just like how likely you are to outflow from the cell by moving either left or right. So we can think about, right, like we want to fit these parameters for like left and right flow. We have this kind of functional form, which predicts the concentration at the next diagonal in terms of concentration in previous diagonal plus these parameters. And so the way we do this is exactly like that. So we have a model that takes the previous diagonal and tries to predict the next diagonal in terms of these parameters. And compare it in terms of just MSE loss with the actual data. Right. So, OK. So it goes kind of diagonal by diagonal. And if you look at like the individual fits, like they're amazing. Like they look exactly like the data, but it's kind of a stupid thing because, well, you use the previous diagonal to predict the next diagonal. Maybe you like didn't even have to do very much. Maybe you can just like average the adjacent cells or something, and you will do a good job. And, right. So, so what does it mean to actually make a prediction in this case? So the way we make a prediction is we actually start with the first diagonal is constant, right? So now we don't use, the only thing we can use is these like left and right parameters that we fit from the data. And so the model kind of unrolls in a recursive fashion from a constant concentration in the beginning. And I feel like on this patch, I think this is pretty amazing. I think you're going to have to look hard to find where it's actually different. It's different. Right, so this is like this nice example patch. And so the way to think about it is like we have this data. And what we're actually just performing is some kind of very like compression operation. So I'm taking this data, I'm saying like, no, I'm going to represent your data just in terms of this. This is much now, very fewer parameters. This is like, this was 2D, this is now 1D. So this is your representation, and I just give you this decoder. And this gives you your data back approximately. So I'll show a lot of plots that look like this. The top's the data, the bottom's the prediction. So in this particular case, they look very, very similar. It's hard to tell which one is which. On like a more real case, you're not going to have so much trouble telling which one is which, because one thing, the data is like very noisy, and this ends up looking quite smooth. But we make a lot of plots that look like this. And so this is the data. This is the prediction starting from a constant and just using these left and right parameters, which are plotted alongside here as the representation for the data. So you can actually get pretty good fits from this model. OK, but actually, this model is like not really, I mean, of course, it's not close to being complete. There's lots of things we're not modeling. But actually, there's a very simple thing that we know is present in the stochastic model, but we're completely missing in this model. And the thing that's present in the stochastic model is when you run the stochastic model, you actually you have more than one cohesin on the DNA, right? And which is realistic, right? People know like what the concentration of cohesins are, and you really should have more than one. And so they have an opportunity to interact. And OK, what happens when they interact? And this again, we take inspiration from Jeff's model, which dictates that when cohesins bump into each other, like they're trying to move past each other, they can't. They become more likely to detach completely, right? So. So we made this what we call like the cohesin stalling model, where actually it has exactly all, doesn't have any more parameters. It just has a different functional form. So it has this kind of implicit stalling term on the bottom. And turns out the stalling model actually fits the data much better. So, and there's an interesting phenomenon where, so a bin is 1K actually, so this is 20K bins. It's actually exactly the same patch, just like at different resolutions. It's a biophysical model, so it should kind of scale nicely to different resolutions and behave exactly the same. But we found that the basic model really actually kind of breaks down at the finer resolution scale, but the stalling model actually can still recapitulate a lot of the data. And in particular, what the stalling model is really, really good at is creating these, like, what I would call like these passive TADs at all the different resolutions. And this is just kind of like an overall plot where we look at like like the very fine resolution and the more coarse resolution and across different cell lines. And overall, this blue stalling model is actually definitely fits the data better. Has no more parameters, just different function, right? So I think this lets us learn something about what the realistic biological model formulation is. And okay, and so finally, I guess these kind of models over here. So we call them like direct fits in the sense where all we're really doing is data compression. It's like not a predictive model at all. Like I've taken this patch and I've like generated this very small representation for this patch from which I can get it back, but I haven't learned about anything else that's happening anywhere else in the genome. Like I can make no predictions, just nothing. Right? So this is the direct fit model, and ideally we would like to use it in a predictive context. And so the way we use it in predictive context is now this is kind of a sequence to function model. So we have a reference genome which has sequence features. So the CTCF positions left, right, and we want to do this in like a cell type specific way. So we have some like a context vector, which is the open chromatin for that cell. And we take these and have some kind of fairly small neural network that just outputs our left-right parameters. And from the left-right parameters, we generate the data so we can compute kind of the error on our data, and this whole model can be fit end-to-end. And now this is actually a fully predictive model. Here's a bunch of patches that this model has never seen before. And OK, so it is not Orca quality or Akita quality, right? Like, no, it's not. If you're like squinting at it and like, is this actually good? It's not that good. But we only used 536 parameters. That's it. And we also didn't, like, we used very little of the sequence, in fact. So like, we had a lot of discussions about data leakage. Like, oh, is this a homologous region that you have seen before? Like, what happens if you left that out? We actually really don't have to worry about this because we literally just use the CTCF sites, and that is it. There's no actual sequence went into this model. It was already pre-featurized in this way. So yeah, this is, I think, like, it's pretty promising. We're working on getting like a full sequence model, and of course, like, one advantage of this model is this is a thing that can just train in a few minutes on your computer. You can really play around. Like, you can play around with the parameters, the model formulation. It just lets you do so much. As opposed to like the full sequence model actually takes some time. So yes, it's more accurate, but you can explore less, and there's definitely a trade-off. And so I'd just like to finish with thanking the people who worked on this project. So the totally interpretable sequence to function model was led by a student in my group who actually just graduated. You want a postdoc? This is Churl. He's amazing, and he's done so many diverse things and really excelled at all of them. And the DLEM model is really a collaboration between Tina, who actually presented the idea for this model at MLCB last year, and Turl as well, who's working more on the sequence to function aspect of it. And I also I like to thank all the other members of the group. All of the great collaborators have been indispensable with their insights and advice, and I'll take questions.
Thank you for the great talk. Very energetic, too. Okay, let's see if there is any question.
Hi, a very cool talk. I was wondering if you looked at if you found sequence features that are present but are missed by your model, like that say there's an absent loop or an absent TAD that is present in the real data that's driven by something that's perhaps not CTCF?
Yeah, I mean, that's certainly like a direction that we are going, and we haven't looked as much in depth into that issue as the reverse issue. And there seems to be like an interesting trend because as you move away from diagonal, you have very few reads. But you know, the biophysical model is really like not bound by that. It still makes a prediction, and so it predicts all of the structure. And I'm like, I don't know if it's there or not. But actually, like people have made kind of like more local data sets that are have more reads. And actually, if you look at that, it is there. So so I think that's kind of one aspect. I mean, and the reverse, like we haven't looked so much at it, but it's definitely there. I mean, right now we're modeling like very, very few things. Of all the things that I know matter, we don't even model all of those, and I know there's more things that I don't know about. So.
Yeah, very nice talk. I was wondering, so how to sort of like interpret these vectors that you get, that you kind of create here with the left and right transition rates. Basically, if you had basically two different conditions, two different cell types, you would get like two different vectors from that. Like if there's like a difference in your IC map, I'm wondering how you would interpret that kind of knowledge that you get now from these this new representation basically of the data.
Right. So yeah, you do. Actually, I don't have any cell type comparisons, but I do have the plots, so I can show them. So yeah, you do get things that are different, and they're kind of subtly different. And I mean, the high C itself is also subtly different. The features are mostly conserved, and you can predict the cell type differences, like in this like sequence way where you just use CTCF and some data like CTCF, like a site whether or not it's present, and you have some kind of cell type specific data that tells you like, oh, is the site occupied, right? So it could be like CTCF or you know, like a TAC-seq as a proxy. And the occupancy of those sites is variable between cell types for reasons that I think are not super well understood, having talked to experts about that. But yeah, so there are places where the cohesion complex can't proceed in one cell type because there's a CTF there. But in a different cell type, it's missing for some reason, and extrusion just proceeds. So you can capture that, a lot of those elements quite nicely with actually this very simple model.
Very neat talk. So with regards to the first project, there was a paper from Wyeth Wasserman's group that I think was presented at MLCB a couple years ago that was suggesting you learn a complicated 1D function related to each motif, but didn't do any of the interaction modeling. I'm wondering if you have comments about...
That's right. I'm not the best with names. Yeah, so I think, I mean, this was almost, I guess, like a similar idea, but like with like completely orthogonal parameters, I would say, right? Like, because the things that we like held constant, they fit, and then, yeah, I mean, I think like I thought that was good work, and I think like trying to make the simplest model possible is super informative, and we should all be trying to do that, and you know, and really trying to understand like what are the super important features to get like, even if you get like 90% of the way there. So, and I think like, I mean, yeah, somehow the main like, in a TAC-seq, like a major nonlinear thing is just the saturation effect, which is like, oh, chromatin can only be so open, like it's open, right? So, but for some things like, you know, some of the MPRA people argued that linear models fit great. So, right, and it's almost like in the, it's almost just like down to your opinion whether or not you want to care about kind of higher order terms. So yeah, I think it's really nice complementary work.
Great talk. I have also one question on the first model, on the interpretable CNN model. It's really interesting to see the saturation effects. I wonder how diverse are, because the motifs are predefined, are they? So how diverse are these motifs in the model, or are there similar motifs? Could that kind of lead to potential saturation effect? Thank you.
Yeah, so our motifs were like cis-BP because we really like almost followed AI tech like line by line, and that is what they used. So in some sense, like the total input to our model is exactly the same as the total input to their model, including the input you need for interpretation. So we actually have a ton of motifs in there, just like way more than we actually need. And in the paper, one thing that we do that I didn't show here is we can make the whole model sparse, so like have a lot of the parameters actually go to 0, which is a little tricky in this kind of like non-convex context. Yeah, just overall, like there's way more, for any given thing there were more saturation interactions, and overall there were more saturation, even most of that is like explained by CTCF, but just like saturation was dominant.
So I guess for interpretability, this is, I guess, a bit of a broader question, but I guess in the LLM space, there is this sort of movement and research direction of mechanistic interpretability, where you take these like really complex models and try and enforce some sort of constraint on them, like sparsity or something, and then try and identify individual neurons that correlate to some sort of features. What do you think are the trade-offs like between that direction and instead, I guess, making a model as simple as possible?
Well, yeah, I guess I don't have a ton of evidence for this, and I think with time we will know more. That's why I kind of started with a solar coast example, like as a kind of inspiration. And I think you can think about like, okay, what would it have taken for pattern matching to succeed at this task, right? Like in the 1600s, you were so far from being able to do that. Like there's no computers, no data, the instruments are all terrible, like everything is terrible, and yet you have like this one nice simple model that does amazing things. Can we have something like that in biology? Because we have all the same problems. Like the data is terrible, actually, right? We don't really have great quantification in any of our assays. And we have computers, but I guess we always want more. And yeah, maybe just maybe, I don't know. But maybe I think you can get somewhere very far by just formulating things in like a general way.
Thanks.
Let's thank the speaker.
Great.